Something else for us to note about the cell cycle is that it is very carefully regulated. And what I mean by that is that there are factors in place that can speed it up, that can slow it down, can stop it altogether even. And this regulation is there to prevent what we would call uncontrolled cell division. These regulatory molecules are found in the cytoplasm of all cells, and they are constantly monitoring and regulating this cycle as it goes on and on through the life of a cell. Now, if uncontrolled cell division occurs, we set up a situation where mistakes could be made that could lead to problems with the organism. And whereas in a normal, healthy situation, a cell like that, that's damaged and irregular, would be prevented from dividing. If we lose control of the cell cycle, that abnormal cell might continue to divide, and then we end up with things like tumors and cancers. I want to spend the next couple of minutes talking about one of these cell cycle regulators. And this is one I'd like you to learn about in detail because it's one of the most well-studied cell cycle regulator, if not the best studied. Again, this is a regulator that is present in the cytoplasm, and it's called MPF. And I'm happy for you to learn it as MPF, because that's what most people refer to it as. But MPF stands for something. It stands for maturation promoting factor. Another term you'll hear is mitosis promoting factor. So this is a regulator molecule that helps the cell cycle move from G2 into M, when M stands for mitosis. MPF is actually a protein, and it's a protein that's consisting of two parts. Two individual proteins that come together and function as a single unit called a dimer. Now, one of the proteins is called cyclin, and the other protein is called cyclin-dependent kinase, or CDK. So one protein, cyclin, joins up with CDK, and those two things together act as MPF. Now, cyclin turns out to be an interesting molecule, but CDK does too, in that CDK is actually an enzyme. And it's an enzyme that belongs to a great big family of enzymes called kinases. A kinase is the type of enzyme that can phosphorylate a protein. Now, when I use that term phosphorylate, what I'm saying is that it can place a phosphate group onto a protein. A phosphate group is a single phosphorus atom and several oxygen atoms. And when you place one of these phosphate groups onto a protein, it changes the protein. It alters the chemistry of the groups that interact on a protein. And in other words, it causes the protein to fold a little bit differently, which changes its structure and thus changes its function. Remember, in proteins, structure is tightly connected to function. If we change, for example, the amino acids in a protein, we're going to change its structure and change its function. Similarly, if we phosphorylate a protein, we're going to change its structure and change its function. So in this slide, what you're looking at is a generic protein in a circle here. And we can see that a phosphate group up here attached to an ADP is being added on to our protein. And that's what's represented by the P in a circle. Now our protein has gone from unphosphorylated to phosphorylated, and in the process it's changed shape. It started off with a circular shape, and now it's become a square. Now, if you were to remove that phosphate group, and there are other enzymes that perform that function, the protein would go back to its original circular shape. Oftentimes, this cycle of phosphorylation and dephosphorylation helps to either activate or inactivate a protein. And the same is true with MPF. It turns out that originally, in its inactive form, CDK and cyclin are phosphorylated at two places. So the CDK cyclin molecule has two phosphate groups on it. That's its inactive state. Late in the G2 phase of the cell cycle, enzymes come in and they take off one of those phosphate groups. So now the MPF molecule has only one phosphate on it, and now it's active. When it's active, the CDK part of the molecule, because it's a kinase enzyme, starts to phosphorylate proteins. It travels around the cell, essentially around the nucleus, and it starts to phosphorylate things. It phosphorylates the chromosomes, and it phosphorylates the nuclear envelope, and it phosphorylates some microtubules and other things. And all of this phosphorylation is preparing for the events of the M phase. Now, the other thing that maturation promoting factor, or MPF, does when it's active is it phosphorylates an enzyme that will turn around and break it apart. In other words, MPF phosphorylates an enzyme that will inactivate itself, degrade itself, and shut it back off. What this makes sure of is that MPF will only be active for a short period of time. It will be activated, it will phosphorylate things in the cell, and then it will be inactivated. And that way it does its job and then it's finished. So in this slide we can see an image that represents active MPF, active maturation promoting factor. And they're showing us that this is one big protein, very globular shaped, that's actually composed of two individual proteins called cyclin, shown in blue, and CDK, shown in green. So these two proteins come together, and normally there are two phosphate groups attached to this, and that would allow this CDK cyclin to be inactive. When an enzyme comes along and takes one of these phosphate groups away, now we have active MPF. And that MPF is going to go around and phosphorylate things in the cell. It's not important to me that you memorize the individual things that MPF phosphorylates when it's active, but I do want you to understand that it phosphorylates things in order to activate them and prepare for M phase. And of course, I would also like you to understand that active MPF phosphorylates an enzyme that will turn around and break it apart, break it back into cyclin and CDK. And of course, MPF is only one cell cycle regulator. It's one that's very well understood, but there are lots of cell cycle regulators, and we're discovering more and more of them all the time. Another very famous cell cycle regulator is called P53. P53 is a protein that has the job of stopping the cell cycle. So P53 is a protein that can put a cell into G0 phase. And a cell would go into G0 phase when it was abnormal. P53 is a protein that can recognize an abnormality in a dividing cell. And so it puts it into G0 in order to stop the cell cycle and help the cell undergo a process called apoptosis. Apoptosis. This is a pretty horrible looking word. It looks like it should be pronounced apoptosis, but we pronounce it as apoptosis. And that process is what we would refer to as programmed cell death. So by programmed, I mean it's step by step by step. It's a normal, healthy way that a cell would take a damaged or irregular cell out of the tissue and destroy it. P53, as you might imagine, is a very important protein in that it can actually help prevent uncontrolled cell division and prevent tumor formation and cancer. For those reasons, we refer to P53 protein as a tumor suppressor. It's a normal protein that's present in our cells that helps prevent the formation of tumors and cancer. Now, in terms of regulating the cell cycle, there are some very specific times during the cycle that the cell takes stock, if you will, to make sure that everything that's been done so far has been done correctly. We call these times cell cycle checkpoints. We have one checkpoint at the end of the G1 phase, or late in the G1 phase. We have another checkpoint late in the G2 phase, and we have a third checkpoint in the middle of the M stage. And that's what you're seeing in this picture. These purple lines represent the G1, G2, and M checkpoints. These again are times when molecules within the cell are able to detect any abnormally replicated molecules. So at the end of G1, if there are any abnormal cytoplasmic components being copied, then that will be detected. At the end of G2, if there's any abnormally copied DNA or other cytoplasmic compounds, those will be detected. And the last one that occurs during mitosis occurs at what we call metaphase. We'll talk about that next week, but this is a time when the cell notices if the chromosomes have been divided equally into two sets so that each daughter cell gets a full, complete set of chromosomes. So, again, late in G1, the cell's going to make sure that the cytoplasm has been doubled and the cell size is adequate, and that the DNA is undamaged. Remember, at the end of G1, the cell is about to go into S phase. It's about to copy all of the DNA. So it's important for the cell to take the time and the energy to first make sure that the DNA is normal before it gets copied. This ensures that this cell that's about to have its DNA replicated is worthy of dividing. If the DNA is damaged at the end of G1, rather than go ahead into S, P53 protein will turn on various genes which will stop the cell cycle, and one of two things can happen. Either the damage can be repaired, if it's not too significant, or the cell can go into G0, stop dividing, and go into this death process called apoptosis. At the end of G2, because the cell has gone through S and had its DNA copied, the cell's going to make sure that that process went well, that the DNA has been copied, that it's undamaged, that all of the chromosomes have been doubled, and it's also going to ensure that activated MPF is present. Because remember, it's MPF that's going to prepare things for the M phase. If there's something wrong at the end of G2, the same things that we saw at the end of G1 can happen. P53 protein can be activated. Other proteins can help the cell go into G0. The damage can either be repaired or the cell can go into apoptosis. During the metaphase checkpoint, remember that's in the middle of M phase, what is checked is that all the chromosomes are attached to what's called spindle fibers. And again, we're going to talk about that next week. But for now, understand that this makes sure that each daughter cell gets the correct amount of chromosomes, or the full amount of DNA that it's supposed to have. When checkpoints fail, and they do fail, Remember, biological systems are not perfect, and so all of these processes are subject to problems. And when the checkpoints fail, what ends up happening is we have a cell that goes into an abnormal division. Remember, if it's uncontrolled cell division, we produce what we refer to as a cancer, a tumorous mass of cells that are dividing in an uncontrolled way and producing a cancer. The other thing that can happen is that you end up with daughter cells that have normal DNA but abnormal numbers of chromosomes. And this, of course, causes disease. Probably the most commonly referred to example of that is the disease that we refer to as Down syndrome. Down syndrome, or trisomy, is a case where a daughter cell gets an extra chromosome. And if that is not corrected, not repaired, then you end up with a person who has an extra copy of a chromosome, and that extra copy is going to make extra proteins that are abnormal, creating this disease syndrome that we refer to as Down syndrome. So the cell cycle has to be regulated, and the checkpoints that are in place ensure that the cell cycle proceeds normally and keeps cell division normal and healthy. When we get together next week, we're going to talk about the M phase in great detail. We're going to talk about the stages of mitosis and what happens during mitosis, and then we'll have our laboratory where we actually get a chance to look at each of these stages of mitosis underneath the microscope in a dividing cell.